This is slide number or video number 2 going from here. So where we left off was animal testing and why we have to do animal testing. And that's because there is no better model, better predictive model, nothing computer-wise that is even close to being a good predictive model of what will happen in humans for a drug. So until we get to a point where computer models are there, it's going to be this way. Right? So this is called preclinical trials. So this is where it starts getting expensive. You're going to need more of the drug. So the in vitro testing, maybe milligram quantities. Now you have to make grams or even kilograms. But grams of a drug, that's 1,000-fold scale up. A lot of the laws, which are called GLP, which stands for Good Laboratory Practice, okay, those laws, they basically outline what the facilities have to be. So animals aren't just in someone's garage. There's laws about how many animals can be in a cage, how often they have to be looked at. They have to have a facility that's designed for animals to live in. So they're called vivariums. They have to have veterinary professionals, so you have veterinarians on staff to do a lot of the work, and animal health technicians to make sure the animals are well cared for, that they get food and water and all that. And honestly, it's good science. If animals are sick because of how they're being taken care of, then you can't really tell if the drug is making them sick either. So it's important that the animals are well taken care of before all this stuff happens. And again, the FDA monitors all of this stuff. And if you're interested, you can look up GLP, Good Laboratory Practice, and you can see what some of those laws are. They are very, very detailed. I might even have some slides on it buried away somewhere on my computer in my office. All right. Now, one thing that starts to get looked at in vivo testing is ADME. ADME is very important. ADME is administration, distribution, metabolism, and excretion. So when you take a drug, how is the drug administered? And also this is called the pharmacology and the pharmacokinetics, which is not only how is it administered, but how quickly. So for example, how can drugs be administered? You could have a drug inhaled. It could be eye drops. It could be injected. It could be something you swallow. You know, the goal, the ideal, the perfect drug as far as people actually taking it would be something you take once a day, a pill that you swallow. Right? Distribution. So once the drug gets into the body, where does it go? Does it go everywhere? Does it concentrate in the lungs? Does it concentrate in the liver? Right? Where does it go? Metabolism. So what happens to it? So your body's goal, as soon as anything comes into your body tries to get rid of it. So what are the chemical reactions by which the drug is broken down? And then where does it go to be eliminated? And that's what excretion is. Is it excreted by exhaling? Does it go out in the urine? Does it go out in the feces? Is it sweated out? Any number of ways. Now, how do they determine this? Some of you aren't going to like this, but what they do is they give drugs to several animals, and then they're going to sacrifice them at time intervals, and they're going to analyze the tissue, and they're going to look, doing chemical analysis of every piece of that animal's tissue to see where the drug is, how fast it got there, so they do different time points. So the idea is that you're going to get as much data out of each animal as possible, but the only way they know is, you got to sacrifice the animal and analyze the tissue. Right? And they want to get the rate as well. And that's important to know. That's how we know how long a drug lasts in an organism. And how often do you have to take the drug? Well, that depends on how long it lasts. All right. So the animal testing goals out of animal testing, you're looking for ADME. You want to get dose ranging, toxicity. And how do you know toxicity in animals? Well, you can tell if an animal dies. But how do you know if an animal doesn't feel well? You can't ask the animal, do you have dry mouth? You can't ask it. But that's why you have veterinary professionals and animal health technicians. You can tell the animal's not eating well, or it's losing its fur, or it's any number of behavioral changes. So there's all sorts of things that people who are trained in working with animals can look for to determine if the animals are not feeling well. But we look for toxicity in animals, right? Sometimes efficacy, because we can't always test, does the drug work in an animal if the animals don't get the disease, right? But if the animals get the disease, then you can test it. Right? So sometimes efficacy. But you're definitely looking for toxicities and doses. At what dose does it start getting toxic? All right. And again, you can imagine what this costs, because you've got to pay all those people. Veterinarians are well paid. all the animal health technicians, the facility, all that kind of stuff. All right, now once drugs get through animal testing, assuming a drug gets through and it shows some promise and doesn't look too terribly toxic, then it goes into human clinical trials. So it has to be shown to be safe, and I did put safe in quotations because safe is a relative term. And there has to be FDA approval to begin clinical trials. I think it's called an IND, an investigational new drug, which gives you permission to give a drug that's not approved by the FDA to humans for clinical trials. There are basically 4 phases. That first diagram talks about them, but there's phase 1, phase 2, phase 3, and phase 4. And you can see that the order of magnitude, the number of people being tested, increases significantly at each stage. Phase 4 is actually aftermarket. All right. So I'm going to go through each of these and what they're looking for. All right. So phase 1 clinical trials. This is, of the order of magnitude 10, so 20 to 100 healthy volunteers. They love college students because college students are always looking for a few bucks. So they get paid. Typically, you're healthy. They don't take women of childbearing age for obvious reasons, right? And typically, it's going to be a short, in a clinic where you'll be in a bed, a hospital bed, and they're going to have tubes. They're going to do blood draws. They're going to monitor your heartbeat, your breathing, probably urine samples. How do you feel every so often? But you get paid for doing it. But the goals, you want to get ADME in humans, right? Now, you cannot kill the person and take tissue samples, but you can take blood every so often. You can take urine every so often. You can monitor heartbeat, things like that. They get dose ranging. So how much do we have to give to these people before we start seeing some toxicity? Now, you already have a feel for it in animals, but humans and animals are not the same, but they can be pretty similar. And also side effects. And of course, a human can tell you if they're having weird vision, if they have blurred vision, or if they have dry mouth, right? Animals can't really tell you that. So that's phase 1. Healthy volunteers. These people do not have the disease. So you're only looking really safety in phase 1. Phase 2, you ramp it up by a factor of 10. Now we use what are referred to as healthy sick people. So they'll have the disease, but they don't have anything else wrong with them. So for example, let's say it's high blood pressure, but you don't have high blood pressure and obesity and diabetes and lung disease. You only have high blood pressure or any number of other things. This is where you first start looking for efficacy in humans. So they have the disease, but they don't have anything else wrong, right? And again, and by the way, these are controlled. So people are getting dosed, right? They're getting the pills at a certain time of day. It's very controlled when people take the pills, how much they're getting, right? And again, the goals, first efficacy, dose ranging, and side effects continuing. All right, and in the first slide, it tells you these things go on for a while. And by the way, it's not necessarily one trial. There might be several phase 2 trials going on at the same time. Okay, then phase 3, and it's clinical trials plural. Now you got people, they can have other things wrong with them. It's much less controlled. You can see it's thousands of people. It's a much broader populational cross-section. And again, efficacy, side effects, drug interactions, because they can have other things going on. Acute versus chronic toxicity. So acute means I take it and I get sick right away. Chronic means I have to take the pill for a week before I start feeling anything, or a month. Consider this, right? Some drugs, if you get on high blood pressure medicine, you might take that for the rest of your life. If you know, those of you who have older parents or grandparents who take 2 or 3 pills every single day, right? Right? But that's the goal in phase 3. Now, some other things about clinical trials. Clinical trials are very expensive. The price goes way up for drug. So the most expensive part of drug discovery and development is animal testing and clinical trials, because now you got doctors and nurses and other health professionals, right? You got lots of subjects. The regulations for this are called good clinical practice. Okay, and also a very big thing is the placebo effect. You may have heard. Placebo effect is you can give them a sugar pill and tell them, oh, this pill will make you better, and it sometimes works. So in clinical trials, that has to be demonstrated that the drug showed significant improvement over the placebo. So you typically have one group gets the placebo, the other group gets the drug. In a blind study, the patient doesn't know which one they're getting. In the double blind, the patient and the doctor doesn't know what they're getting. And that's because the placebo effect is very powerful. And then they just have numbers, and the people who read the data can decode the numbers. Another big deal is what's called informed consent. Informed consent means if you're a subject in a clinical trial, you understand, you're consenting and you understand what could happen to you, what the test is about. You're saying it's okay, but you're well informed. And there's a lot of laws about what constitutes informed consent, and that came out of a lot of things that were done. The Tuskegee experiments in this country, where they let some people just die of syphilis to see how it was going to happen, right? And they didn't know. So that's what informed consent is. It's a big deal, and like I said, there's a lot of laws around this very, very specific things. All right. Phase 4 is aftermarket, and it's much less controlled. One of the big issues in just drug safety is patient compliance. Patients, the biggest issue, frankly, with drug safety, if you ask professionals, is patient compliance. They don't take the drug when they're supposed to. Oh, I'm going to be busy, I'll just take 2 now instead of taking 1 every 4 hours. Or I'm supposed to take it until the pills are gone, but I stop after 3 days. I mean, some of you may be nodding your head, right? It's very important to follow those instructions, because the drugs were designed based on, or those instructions came from a lot of this stuff. Anytime a drug causes any kind of an adverse effect or an adverse event, there's always a number that you can call. They have to listen to and follow up on all those complaints, because once it goes out into market, you're going from thousands, millions of people may be taking the drug. Right, I'm going to stop here just because I want to keep the videos a little bit light. So we'll stop here. Oops. Go to the next slide, sorry. Okay, stop here, and then we'll go to the last set of slides. Let's finish clinical trials. We're almost done with these.